Okay, welcome back. We are ready for our next speaker. Please welcome Yael Gozin from Pfizer, presenting Quality Transformation Initiative.
Okay. Okay. Good afternoon. I know that most of the sessions here are, I'm presenting and you sit silently and listen to me very carefully, of course, and ask questions at the end. My request is I'm going to present a very specific use case. So, if you have questions during my presentation, please stop me and ask. That will be way more fun and interactive. And of course, any questions after or, in a week or whatever, I'm more than happy to answer. So let's start with Quality Transformation Initiative, which is a name that we gave to a pilot, which we were trying to do. It's very fancy and we like it, but it's also very specific. So when we talk about, and I'm going all the way now to the end game. So we were talking about, basically regulatory submissions. Regulatory submissions at the moment when the problem is that the amount of the data that we're submitting is growing, and it's growing basically exponentially. If you look at the size of the submission and the complexity of the submission from even a decade ago to today, the number of basically pages that are submitted to the FDA and the amount of data is basically much, much bigger. The problem is that the processes around compiling these submissions and around checking the submissions, so basically verifying the data in there, it stays the same. We are still doing it manually, mostly. I mean, unless anyone here doesn't do it manually, but most of the industry is still doing, the processes are pretty manual, they're laborious, they're time-consuming, and unfortunately, they're also error-prone. Now, the question is, okay, so now we need to find a solution to make it, first of all, probably because it may impact the quality of what we're submitting, we want to find a way to improve the quality, but also because of the time it takes, we would like to get, at the end of the day, the goal is to bring the drug to the market faster. So we would like to find a way to speed up some of these submissions timelines, and not by putting another 20 people to check the numbers, but doing something which is more reliable and sustainable and scalable. Because we definitely don't know how these submissions would look like, for example, in 5 or 10 years, okay? So just out of curiosity, one of the things that we did, we looked at the size and the number of these submissions in 2018, so last year. Last year, the number of submissions or applications that went to the FDA, so it's everything, safety, new drug applications, and any type of supplement. We're talking about around 200,000 applications. Out of them, the rejection rate is pretty high. The rejection rate due to data issues, and data issues is a big word, was more than 21%. So it was actually 21.6% here. And we're talking about inaccurate dosage that was reported, the inaccurate number of patients. So things that we basically should have seen, and we didn't because we submitted millions and millions of pages for the FDA to review. If you're interested, actually, there are these references to where these numbers are taken from. Now, so then the question is, okay, can we make this process reliable? How can we make this process reliable? And of course, as I mentioned before, we were talking about automation. Now, automation of this type of data. So what we need to do, we need to basically verify data point against source. And in my case, and I will talk about it later on, we're talking about endgame, so we're talking about submissions, but this concept of verifying structured data in healthcare is not as simple as one might think because of, and I think that was mentioned throughout the presentations today, the data is in different formats, the data is in different structure, the data is in different standards, but even if we look, and I'm smiling because even if we look at our own documentation within the company, it's enough that you have different medical writers who are trying to be slightly more creative, and if you want to compare source to document, you're lost because you cannot even put enough set of rules to address all the variability that you have. And we tried. I have to say that we tried. So the original attempt was really to use rule-based NLP, and we failed miserably because we couldn't come up with all the impossible set of rules that was associated with what they needed to do. And then, of course, the word AI, or specifically machine learning, came into, into everyone's, I don't know, like knowledge, and it became such a big buzzword. So we said, okay, can we train a machine to help us? Can we train the machine on the variability that we have in the data and try to address that? So we built a very specific use case. And again, every time I'm presenting it, so forget the fact that in this case, it's a very specific use case that talks about documents and program tables. At the end of the day, after this presentation, just remember what we're really trying to prove here is that we basically can use NLP plus machine learning in order to verify structured data. Now, if you change the use case, of course, you have to retrain, but you definitely can leverage the things that you already did in order to retrain. And unfortunately, we have to retrain, and I will show you why. But again, the big question is, can we verify structured data? And that's what we were trying to do. So again, the objective was to demonstrate that we can automate cell-to-cell verification in a clinical study report. And basically what we were trying to do, we took a clinical study report, big part of it is basic tables. And that's a Word file, I think, in most organizations still. And we have program tables, SAS program tables that come presented in different formats. And what we were trying to do is basically to automate this process. Instead of people sitting and comparing the tables to tables and numbers to numbers, can we automate that? So how was it done? We partnership with a company by the name BeaconCure. Now, BeaconCure is a small startup. It's a really small startup. It's 8 people startup. And what we did with them, we developed a cell matching methodology, which is basically what we wanted to do is, again, structured data verification. Now, an obvious question to ask me, why didn't you use an existing platform? Why did you just went and decided, because we have platforms that are using NLP, we have platforms that are using machine learning. Why did you decide to go and develop something from scratch? We actually tried, when we started, we tried to use an existing platform for one of the big tech companies. And we decided to give BeaconCure the challenge and say, okay, can you develop for us algorithms from scratch? Can you work with us and develop the algorithm from scratch? They did much better. They did significantly better. So when they understood the problem, they worked closely with us, and they were not bound by the limitation of their own platform, because there was no platform. They developed everything for us. So that's why we continued the engagement. So at this point, we developed this. The idea was to develop the methodology. So they developed the cell matching methodology. And the whole idea behind the methodology is basically each data point in the table has a unique characterization. And I will talk specifically what this characterization is, but think about it. It comes with a lot of metadata that attach to this data point. So what they did, they basically transferred the characterization, the unique characterization, into a database. And then when the information was in the database, they did the matching. So what they did, they didn't match the values. They basically matched the characterization. They said that, okay, this characteristic is equal to this characteristic, so now we can look at the numbers. And just to illustrate it a bit better, and that's basically the essence of everything that we were trying to do. So if you look, oh, excellent. So I'm going to show this part, but it's the same on the other side. So this is basically the document, and you can see that these are how these documents look like. It's a Word file, and the source is, it doesn't work. Okay, so you have the document and you have the source. Now, I mean, in this case, the document and the source are basically look like they're verbatim. So the headers and the columns are like the titles are verbatim. Many times, I don't know if you saw clinical study reports, the medical writers will many times amend it a bit. So it will be, of course, the same meaning, but it doesn't have to be verbatim between source and table. And that was one of the challenges when we had to train the tool. And now each one of these cells, so for example, we can look at the document. So each document has, let's look at row 1, column 1, and it has a number of attributes. Now, again, that's an illustration, okay? There are much more attributes, there are more defined attributes, but in this case, for example, each number, you know where, from which document it came, from which table it came, from what was the title, what was the type, the headers, the row headers, the column headers, and so on and so forth, okay? And the same thing can be done for each cell in the source. So for example, in this case, again, row 1, column 1 for the source. Again, there is a list of attributes that associated with a specific cell. And of course, you know, we can look at another cell. And in this case, again, there is a specific attributes that are associated with the other cell. And you can see that they're basically, you know, almost the same. So the only difference between row 1, column 1 and row 1, column 2 in the source, in this case, will be the row header, okay? So, and now the next step will be, okay, we transferred all this information. We did the same thing for each and every cell, both in the source and then in the target document. And we transferred everything to a database. Now what we were asking the system to do is, please find me the matches. Please find me like the cell, if I have a cell in the target, please find me the matching cell in the source, okay? So the first part will be, for example, in this case, if I'm comparing row 1, column 1 to row 1, column 2, the system will tell me, listen, there is a mismatch here, okay? The attributes are not identical. And therefore, if I have a mismatch in the metadata, the system will look for something better, okay? Look for a better match. So in this case, for example, the attributes are identical. And then the system will say, OK, that's great. Now let's look at the values, OK? Because I'm looking for mistakes. So let's look, OK, now I found the right cell. I found what I need to compare to what. Let's look if the values are identical. So what I will see, in this case, 56 and 56 are the same number. So the system will move forward. Now, if when I was actually writing this clinical study report and I made a mistake in transcribing the data, okay? And instead of putting 56, I put in my target report 78 or any other number which is not 56 for that purpose, the system will flag that as a mistake, okay? So if you will think about what I described right now, it's basically what human will do. If I have to compare two documents to each other, that's exactly what I will do. I will look at what is the right match. I will not look at the numbers. I will look at what is the right match to what, and only then I will compare the values and flag the mistake, if it's a mistake. Okay? Any questions so far? Okay, so far so good? Good. So now we had the concept. We had a great idea. We wrote the algorithms, but now what we needed to do is basically to train the system. And here the fun started, because when we train, we need to train, as we said before, one of the things, even if you have the data, you need to train with the right data. And you need a certain amount of data to train. Now, the CSR structure, thanks to ICH, changed in the last couple of years. So we had a limited number of CSRs with the right format. Now, the other thing was we needed to train across therapeutic areas, because if I will train only on oncology, I'm limiting my tool to understand the ontology for oncology only. So the goal was, okay, can we train across the board? Can we use more CSRs from across the therapeutic areas? The other part was to get all these CSRs and get all these tables, because even Pfizer, which is a big company, obviously, and have a lot of databases and all these things across the board, we actually had problem to get the right data, the right dates, the right information, and be able to train correctly. So we eventually managed to do that, which was really exciting for us. And what we did, we trained, so these are the numbers that we train on. Basically, the more you train, the better it is, but that's obvious. So what we did, we trained on approximately 7,500 CSR tables. So obviously, each clinical study report has more than one table, but our unit was the tables themselves. And we had approximately 80,000 source tables. Because most of the CSRs are coming, clinical trials come with the tables that are in the document, but also a lot of supporting information that is submitted as well. And for us, it was a good thing because we could train, again, we had more information on structure of the source and of course, the ontology. So that was a good thing to do. Now, I specifically put the formats here because obviously different formats need to be slightly retrained. So what we did at the time, clinical study report are in Word file, at least in our case. At the time, the source tables, the SAS generated program tables were in a format which is, it's called RPT. So I don't know how many of you are familiar. It's basically a text file, okay? Just to make it very, very, to simplify it. So we trained. And then what we wanted to do, because it was a proof of concept, so we wanted to do it pretty fast, and we wanted to do it pretty cheap. So because we wanted all these things together and we wanted to work, what we did, we also limited the scope. So what we said is, okay, what we're trying to verify is the numbers in the table. So data point in the table. We didn't look at headers, we didn't look at footers. We really wanted to define, we wanted to show that we can do it, okay? And of course, the more, if you want to extend the scope, and we were working on extending the scope, but just to be clear, if you had a dosage form somewhere outside the table, or you had any additional information that is relevant outside the table, for us it was out of scope, okay? So now we run the wet test. Remember, these are algorithms, okay? At this point, we don't have UI, we have nothing. We have algorithms that were written by the company and Excel spreadsheet that were given to us for analysis. So what we did, we ran the wet test. So we gave them 19 CSR tables, okay? Again, same format, and 554 source tables, and we were hoping for the best. And the results were actually way, way, way more promising than any one of us actually imagined. And it was checked and rechecked and rechecked. So what we did when we designed the testing, we took these 19 CSR tables, we introduced mistakes that we knew, we kept the mistakes to us, and we sent it to the vendor and told them, please run it and send us the results. And then we started, so all the analysis was done by us, all the analysis was done by Pfizer, not by the vendor. So I went through thousands and thousands of Excel spreadsheets just to see all the numbers. So out of the 102 mistakes that we introduced, the system could identify 101 mistakes. The one that it missed was a missing parenthesis. We didn't train on parentheses. Now, we also didn't train on plus minus, which we discovered later on. And now the other thing is, out of, if we're talking about false positive, false negatives, out of 570 cells, because the tables have cells, so out of the 570 cells, 557 were, basically validated without any issues, without flagging, without anything. There were additional 13 cells which were flagged, which was, you can look at them as false negative, right? False positive, sorry. But the interesting part was that even the cells that were flagged by the system, when we looked at each and every cell that was flagged as, and it was not a mistake, it was just flagged as by mistake, we could understand why the system did what it did. So, it was clear what was the problem, and it was clear that we can train in the future properly to address this issue. So it was not random. One of the things that we were afraid, it will be totally random and we don't understand why the system behaves the way it does. We could basically look at each and every cell and understand exactly why it was flagged. And in some cases, honestly, I was joking that in some cases the system was smarter than me because it also catched one or two mistakes that we didn't introduce and were there, and that our QC reviewer missed, as great as they are. So just for anyone who is more understanding, better AI than me at least, so just for the numbers, the precision was 0.99. Again, 1 is perfect. We're almost perfect. Recall, 0.97. The overall F1 score was 0.98. So very encouraged by these results, we said, okay, next thing is to build some sort of a system around it and try to get from the users their input. one important thing, and I will mention it at the end as well, but we worked with the SMEs regularly. So it's not that we developed a system and then gave it to the users. We definitely worked with the SMEs and our QC groups to understand exactly what they're doing, how they're doing, and we did get their feedback on a regular basis, which was really important because none of us is really doing this job on a daily basis. But at this point, we wanted to develop a UI, so user interface, so we can upload the information, we can check, we can understand how it's work. And the system came up, we call it Verify, or the BeaconCure call it Verify, and we agreed. And version 1.0 was launched in April this year, and it was supporting RPT format. And then happens what usually happens in big pharma. We were reorged, and on top of being reorged, regulatory, so FDA came in already a couple of years before, but FDA came with the requirement for CDISC. The problem with the CDISC that it changed the format of our output. So no longer RPT. From now on, all the output is in HTML. Do with it whatever you want. Okay, so the next step was, of course, version 1.1 and version 1.2, where we were retraining the system to understand HTML, so be able to parse HTML, okay? Now, but because the results were pretty good, and we tested already, we were convinced that it works on RPT, and we wanted to bring it to life. We said, okay, let's retrain the system. Now, retraining was challenging. Okay, it was challenging because we need to retrain, but the other thing is the amount of data. So at this point, we collected every CSR that was in the right format and had HTML tables so we can train on. But remember, CDISC kicks in only recently, okay? So we didn't have a huge amount of CSRs to train on, so we did what we could. And the result, I will show the results in a second, but again, I think that it's important because when you work on these things, it's a constant challenge that you don't always expect. So we trained at this time on almost 860 CSR tables, and we had around 3,200 sources, and we conducted the analysis. Now, the release was mid-October, so recently, very, very recently. We were counting the cells and the numbers until basically last night. So the numbers that I'm showing you are pretty initial analysis, okay? We trained on HTML, we also trained on PDF. Again, this is very, very preliminary. We're still checking the numbers. BeaconCure basically the company sent me their own numbers that I'm comparing to mine this morning. So I was on the call with them this morning. Okay, I don't understand how you got to this number. So we agreed on the numbers, and that's the numbers that I'm going to present. But I think the long story short is that we are hitting more or less the same precision, okay? So you can see here that, again, the precision is 0.99, recall 0.95, and F1 score is pretty similar to what we have demonstrated previously. Again, it's still a subset. We probably need to train better. We saw things, as I said, for example, we didn't train for plus minuses. Happens. We still don't look outside the table, but we are there. So we are getting there, and obviously the more we train, the better it is. So just to show you how Verify looks like, and that's specifically, I was asked by BeaconCure to show you how Verify looks like, so please do. So you log in, obviously, and now the system presents basically the projects that you are running. So each project is a document that we are verifying, and we can drill down. So if you will open, for example, like we look at project 3. So if you look at project 3, we can drill down here, so you can see all the tables. The system basically analyzes what is the table and list all the tables in the document. And then if we drill down even further, you can see how each tables look like. So the system, if everything is great, it's just white, okay? If the system flags something, it will be in this pinkish color. Now, the user will go and check, the cells that the system flagged. If I agree with the assessment that there is a problem, it will mark it as this in gray, and will note that you need to check it, you need to do something with that and correct it in the original document. And of course, if it's a false false negative, basically, then the user says, listen, it's okay, it's no problem. The system will check it, it's fine. Okay. Now, one important thing, and then you can get a report out of the system as well. Now, one important thing, we didn't integrate it into our writing tools at this point, so it's a standalone system. Any proposals to integrate it, put it on top of when people are writing, it's all great, because at the moment, it's an additional step. But just as a proof of concept and the idea that we can actually don't do it manually, that's already for me, it's huge. Okay, so just conclusions. I think we utilized artificial intelligence capability, so basically NLP and machine learning in partnership with BeaconCure, and we could develop a new method to verify cell-to-cell structured data. We used the stepwise development approach to address both new regulatory requirements, but also internal changes in the internal processes. We discussed the results of this. I just want to leave some time for questions. So we discussed the results of both the proof of concept and the pre-production pilot, or the next step. What is our goal? And this is just the highlights, but the real goal is to be able to verify all the data, all the structured data that we have in drug development, basically from the point, or clinical development, from the point that we have the data comes into the databases up all the way to the submission. So basically not only at the end game, but really can we clean up our databases, not doing it in a manual fashion. And of course, in order to do that, there are a couple of highlights, which is at the moment we're checking data point to data point. We want to be able to check cross documents, so not only one-to-one, but one-to-many. And the other thing is, of course, we want to go upstream. Can we verify not only looking at documents, but can we go and understand how the SAS database are built, and can we verify those? Okay. So lesson learned, I mentioned a couple of those. Most of them sound really trivial, but every time you kind of come across those, you say, oh, right. So, of course, standardization helps. Even though we're using machine learning, the more standard what your organization is producing, the better it is. It's easier to train. Okay, so if I have less creative tables across the board, it's easier to train with less creative tables. Be sure that your end user is aware of what you're doing, is part of your development, is part of your efforts. First of all, because they know things that I don't know, and I think that their contribution was really, really important. But also because they feel part of the process, so it's not an additional thing that we're throwing at them. And someone said in one of the meetings this morning that we want to ensure that IT is not something that stands in our way. IT is part of what we're doing. IT is part of the business. Digital is part of our efforts. Yeah, we are ready to accommodate changes. Again, access to data is crucial, and that was discussed here in multiple meetings, that we need the right data to ensure that we're training correctly. Develop process around different engagement models. So we worked with a startup, and that was one of the first times that at end development game we were trying to work with startup. And it requires slightly different contracts, and it's slightly different engagement form, and different way that we tested the company as well. So it's something that we are learning by doing, but just be aware that at least big pharma is used to work with big tech companies, and that was a slightly different animal. So just keep it in mind. And of course, moving forward to digital age, as we all know, it's slow, but we will get there, and it definitely will be rewarding. So I'm leaving you 30 seconds for questions. Yes?
So you mentioned 200,000 submissions that go to the FDA.
Yeah.
Pfizer is obviously not all 200,000.
No.
Has there been consideration, are there considerations of trying to expand this for the competition for the FDA and have that, become the leader in that space?
So that's a good question. So here's the thing: we're working with our partner. we developed the tool with them, but they are not necessarily our, they don't belong to us. So they developed the tool as a tool, and they probably will be happy to work with other companies as well. Okay, and at the time, I had the opportunity to talk about it with the FDA. I think they showed interest. I know that BeaconCure had a chance to talk to the FDA. But again, these things takes time. So one thing I'm presenting here, and I hope you think it's cool, if nothing else. But the other thing is to go to your individual organization and say, okay, now we want to try it, or now we want to do it, because it's something that is slightly not familiar, and it's in the early stage, right? So you still need to verify that the system works correctly. The idea of 80-20 in these cases, it's a bit problematic. Okay. Can you elaborate? This initiative didn't work when you attempt with third party. Some pitfalls learned. Okay. Yeah. So when I think that the problem was, it's not a third party. We were looking for the right vendor. Okay, and what we were trying to do is to decide whether we want to take a risk and work basically with a startup that we don't know anything about, and I just met them because I was visiting a VC and was asked to evaluate them, or I'm working with someone who is very established, that has a platform, that did similar things in the past, not for pharma, but for someone else, okay? Personally, my biggest, it was my assessment or the organization assessment, and I don't think once it's correct for everything. It was with the specific vendor that we were trying to work with. I think that it required so many adjustments to their platform. If the platform was defined as we are not doing any programming, any additional programming, or any real adjustment to the platform, and just sort of take it or leave it, and we will try to tailor whatever you need to our platform, that's where it's failed. Because again, pharma the data is more complex. Healthcare data is more complex than, for example, banking or insurance. It just was too complex for them, and their ability to change basically change their platform accordingly. Okay, so I don't see the second question, but again, thank you, everyone. I'm happy to answer any questions. I'm on LinkedIn, I'm on the app, I'm on everywhere. My name is pretty rare, so you can find me. Okay, so again, thank you all.